topic, this title, because I promised Garrett that I would come up with something really, really pompous. And so that's about as pompous as it gets. I'm going to try to break it down into English terms for you starting now. The reason I study the sense of taste is because of the inevitable sensory-motor transformation of taste information. You can take your rodent or whatever and give it a visual stimulus, and there are a lot of visual stimuli that rodent's brain will respond to, but that are totally artificial and have no impact on that rodent, that are arbitrary. Same with an auditory stimulus. You can study, if you want to go simple, you can study the response to a sine wave. If you want to get fancy, you can do a fancy torque. But the fact is that the animal doesn't give a shit about those stimuli, and while its brain responds, it doesn't have anything to do with it. That's never the case with a taste. When you have a taste and you or your rodent are experiencing that taste, the stimulus is inside your body, and it is required that you have a reaction, that you actually produce a behavioral response, and those behavioral responses basically break down into a binary decision. On the one hand, if you have something sweet put in your mouth, this is the work of my very first graduate student, Steve Grossman, and so you can see on this female rodent, you can see the forepaws, you can see the snout, and the all-important mouth. And what you can't see is that Steve has just put a little bit of sugar water into her mouth, and what she does is she produces the rat yum face. And this is, the tongue is sweeping around inside the mouth to pull the fluid back in preparation for swallowing it. The rat has made a decision that it likes this. Contrast that with what happens to the same rat if what Steve puts in her mouth is quinine. Now, quinine is an incredibly bitter taste, although it is delightful with gin. And when you put that in the animal's mouth, you get an entirely different response, a different behavioral response. The most notable one is referred to as a gape. It is a large yawning face. You'll see the teeth exposed. What you won't be able to see is the tongue rolling forward to expel the offensive fluid from the mouth. But it looks basically like that. And there's another one coming up. This is very slowed down. This happens on about a 4 hertz rhythm. And then you could see there was a little limb flail as well. That's the rat yuck face. And I'm going to focus most of my talk on this, because this is what I am. I'm a behavioral neuroscientist, and this is the behavior I'm going to focus on for the purposes of this talk. Why? Because it's a decision that the animal has made. And I didn't have to train the animal to make this decision, although I can train the animal to change its decision. And furthermore, it happens on average about a second after the taste is in the mouth, which means that there's plenty of time to analyze neural activity leading up to that. You're going to see, though, that there's a huge variability of that because this is an animal, not a machine. But this is what we're going to be studying, and we're going to talk about how you get from taste to that. And so just so you know, this is not a one species thing. This is a human infant that just put a sour lemon into his mouth, and not unlike my rodents, this human is now making a gape. I'm often asked, what's the difference between rodents and humans? And the answer is not very much. But one difference is that my rats are smarter than this human. Yeah, that as soon as the cameras clicked, I imagine that this infant put the lemon back in his mouth. And just so you know, it goes all the way across to other parts of the phylogenetic tree. Here is a video from PBS on the Kalahari Desert about toads. And a toad has a very tiny brain, although it is a vertebrate. And you think of them as very innocuous critters. The worst thing they might do is pee in your hand. But if you're in the Kalahari Desert and you are a beetle, these are dangerous, rapacious hunters. And so the beetles in the Kalahari have in general evolved wonderful defense mechanisms. The hero of this video has developed this defense mechanism. It tastes like crab. And so watch what happens when, as that beetle rolls along, this toad tries to swallow him and makes a toad gape. And so even any animal that has a tongue does these behaviors. These are not only ubiquitous to the stimulus presented, they are ubiquitous across species. And so that's what we study. And so we are electrophysiologists in my lab, and I'm happy to have debates about electrophysiology versus imaging if you'd like. I think what I will hopefully prove to you in the course of this talk is that, at least for what we're looking at, imaging isn't fast enough to capture what happens. We do most of our recordings, and most of the talk is going to focus on gustatory cortex, primary sensory cortex, and taste. And and we're going to look at how this process of getting from taste on the tongue to making a response unfolds through time. And what we believe in my lab and what I'm going to show you in this talk is, for starters, time matters in neural taste processing. And now everything that I'm going to tell you is stuff that you all already know, and yet we behave as scientists as if they aren't true. And so, for instance, if you have just a neuron and it fires an action potential and it fires an action potential, then there's a pause and it fires another action potential. That means something, but the way we tend to analyze it is we just say, that's 3. And so how does a neuron respond to this stimulus? 3. And we're going to tell you that that's the wrong way to look at it. And that, in fact, what I'm going to show you specifically is that cortical taste responses evolve across a second to a second and a half, and that evolution is meaningful. You can see the process occurring by paying attention to it. I'm going to then move on to another thing that you already know, which is neural ensembles work together to process tastes, but I'm going to get very specific about it. I'm going to show you that single trial dynamics, single trial responses to tastes, are coherent across ensembles, and they are nonlinear. And one of the great things about it that I'm going to show you is being able to get a hold of that single trial dynamic enables us to show that the coherent nonlinearities drive behaviors in real time. And finally, it's not just that neural ensembles work together to process taste. Neural ensembles between brain regions work together. And so I'm going to show you some new data showing that amygdala, in particular basolateral amygdala, couples with cortex into a single processing unit, enabling those nonlinear dynamics. So that's the point. That's the whole talk. I'm going to spend only a very little time trying to convince you of this, although, to be honest with you, number 1 is basically the entire talk as well. So let me get that out of the way by showing you what a cortical taste response actually looks like. This is the overlain peristimulus time histograms, or PSTHs, meaning peristimulus around the time of delivery of the stimulus. And so time going through time, and histogram of firing rates. And so we've taken the responses to all of these tastes for this one particular neuron and just laid them on top of one another. And what you can see from looking at this right away is that it's not as simple as the neuron turns on or the neuron doesn't turn on. It's not even as simple as the neuron turns on to this degree or to this degree or to this degree. In fact, way back in 2001, and one of the things I love about presenting this slide is it allows me to take my 5-year postdoc at Duke University and reduce it to a single slide. And so what you find when you do a whole lot of analysis on this is that, in fact, these responses basically have 3 parts. There are 3 phases to them. The initial thing that happens when the taste hits the tongue is that some neurons do nothing initially, but those neurons that do something fire a burst of action potentials that is completely non-distinctive for taste. And so we refer to this as the detection period. We are not making claims at this moment about what the rat detects at that point, but we as scientists can look at a neural ensemble. We can see that the taste has hit the tongue because of this detection response, and you can't tell anything else from that detection response. But you can see that over the course of just the first couple hundred milliseconds, these responses diverge from one another. And then you get into a new period that we refer to as the identification period, where we, with even very simple analyses, can tell you what the taste on the tongue is. And so we can look at this activity, and sometimes we need a few neurons to do it really well, and we can say which taste is on the tongue on the basis of what the firing rates are during that period. But then you can see that the responses change again, and now you get into a period where the responses are not only taste specific, they are in fact palatability related, which is to say you can now answer the question from looking at these data, what's the animal going to do about it? Does the animal want to eat it or not? And so with this particular neuron, you can see that the strongest response out during this period is to the bitter quinine, and the weakest response is to the yummy sucrose, and the different sodium chloride stimuli lay out in the right way. So we sometimes see the opposite pattern. But either way, a very simple analysis you can do, you can go ahead and you can correlate the firing at any particular moment with the palatability of those tastes. So we can take the array of firing rates at this moment and compare it to the array of palatabilities of the taste, which the animal likes the most, which the animal likes the least. And when you do that, you can see that for the first half second, even a little bit more, there's nothing. The neuron is responding in a taste-specific way there, but there's no correlation to palatability. But then as you go out past the second mark, this correlation increases. That shows you that it's a palatability related response. And for this neuron, it becomes significant at about 0.7 seconds. All right. And so in case this doesn't convince you that what's going on here is that these neurons are processing something out to get to a palatability related response, recent data, I love putting unpublished data in my talk. And so this is unpublished data from my senior graduate student, Kathleen Magler. And so the unpublished data is a little rockier, but basically what she did was she noted the fact that rats, like humans, have individual differences in what they like. And so, for instance, she gave 4 different rats, both sucrose and sodium chloride, both delicious. And what she saw is that one rat much preferred sucrose to sodium chloride, another rat preferred sodium chloride to sucrose, 2 others were more hovered around liking them the same. She also took rats and gave them both quinine and citric acid, bitter and sour, both tastes the animal doesn't like. And again, while it's pretty much the case that quinine is the worst, there are some animals that dislike them equally. And there are some animals that only dislike quinine a little bit more. And so all that Kathleen did is something that we should have done 20 years ago. She actually took these animals that she'd gotten the individual preferences from and then did the recordings. And what she found is that here is the growth of palatability coding across from about 500 milliseconds, peaking at a second. And this is when we use the canonical ranks, meaning sugar best, salt less, sour but less, and bitter worst. And when she instead uses the animal's own performance, she gets better prediction. So it's not so, and so to put this, and to take the whole data set, when you get out to that late epoch that codes palatability, you get an improved performance from using the only animal's own data. So this late period that we get out to doesn't just code palatability, it codes what is palatable for the individual rat. Yes?
When does the motor response start on this axis?
I'm going to talk a lot about that. The motor response typically starts around here, but I'm going to give you much more specific data to that in just a little while. Okay, and so that is all I'm going to do, just plain and simple on temporal coding, although again, temporal coding plays a role in everything else. What I want to focus on next is another thing that, in addition to not using individual animals' data, behavioral data, is another mistake we were making, which was we were looking at this and saying, oh, isn't that a beautiful ramp? Isn't that beautiful how palatability emerges across half a second, a little bit more? And then we decided to look at things a different way, and we discovered that that was entirely wrong. And so the different thing that we did was we decided to take ensemble coding very seriously. And so what I'm going to show you now, PSDHs are basically a bunch of trials of a taste, all collapsed together for a single neuron. What we're going to look at now instead is a single trial where we're looking at an ensemble of neurons. And so here's an ensemble of 10 neurons, and here is their response to a single trial of quinine. Each one of these hash marks is an action potential fired by that neuron. Now, I would imagine that to most of you, this picture right now looks just like noise. And so I'm going to help you with the way our analysis helped us and tell you that right about there, which is, by the way, around the middle of the time period when supposedly palatability is coming on, there is a change. And it turns out that that change is sudden and coherent. And it's so coherent, here's a neuron that basically only started firing immediately after that moment. Here's another neuron that also started firing just after that moment. Here's another one. Here's another one. Here's another one. 5 of these 10 neurons, all at this time point, suddenly increased their firing, suddenly. And by the way, if that's not amazing enough, this neuron suddenly decreased its firing at that time point, and so did this neuron. So the majority of these neurons that we were recording, and this was back when we could only record about a dozen at a time, change their firing suddenly at this time point, suddenly, no ramping, a very sudden change. And so to really analyze this, because I didn't just put this line here by eye, we actually did an analysis that is referred to as hidden Markov modeling. At this point, lots of people know about hidden Markov modeling. Let me tell you, when we first tried to publish on it, they did not. And so hidden Markov modeling is designed to find sequences of states in your data. Up until very recently, if you talked on the phone to a machine and it was interpreting your voice, you were probably talking to a hidden Markov model that was trying to say where the syllables and syllable boundaries were in what you were saying. In the case of our neurons, what they show us is that essentially there is a state here and a state there. the state defined by the population of firing rates, and that in between the two, there is a period in which the analysis is uncertain, but that's where the transition happens. And so in fact, you can, in this analysis, you can basically pull out what amounts to the calculated confidence that say this state exists, which is 0 here, rises really quickly, and is 100% there. And so that's what hidden Markov modeling does for us. And so I'm going to use that now for a lot of stuff. I'm going to actually talk to you about what those states are. But first, just to get it out of the way, let's reconcile the PSTH and HMM-based analyses of taste responses. When we take a random animal and a random ensemble of neurons, and we take, say, 10 trials of quinine, and we throw up on there the likelihood of the state that is dominant somewhere out here. And so this is the state which we're going to refer to again as the putative palatability state, because it occurs out there when we already know that palatability information is high. And so we just pick, I think there are 10 trials of, and said, how does that, the likelihood of that state evolve? And what you can see is that on every single trial, it happens suddenly. And every single trial, the probability of that state emerges all of a sudden, but with a wide variability between trials on exactly when it happens. And so if you just do something simple, like average across those trials, you get that. And that is a ramp that looks decidedly like this ramp. And so we began thinking that perhaps the idea that palatability coding emerges across this half second is an artifact of collapsing across trials in which palatability coding emerges suddenly, but at different times on each trial. And now I'm going to prove that to you. And so this is the work of ex-graduate student Brian Sadaka. And what he did is he collected a new data set from gustatory cortex. And first thing he did was he just ran it through the typical analysis so that you can see that, like I've shown you before, across the population of neurons, you go from being a low correlation with palatability at around 0.5 seconds, a time at which the neurons are coding tastes, and it gets high by a little after 1 second. So this looks exactly like the data we've shown you before. And I will say I've been doing this now for 22 years. I still breathe a little sigh of relief every time a data set comes in and it looks like this. And so then Brian decided to do just a very simple analysis. He took every trial that was used to make this, and what he did was he did hidden Markov modeling on it. And so this is a schematic of the hidden Markov model solution. Each one of these lines is the probability of an individual state in the across time. And so this is based on the ensemble of data. And then he made a simple assumption. The time period out here is where palatability is high. And so I'm going to call the state that is most likely down in that time period, in this case, the one in blue, that is my putative palatability state. Everyone still with me? And then he said, okay, let's just do something simple. Let's find the time point at which that state in each trial becomes maximal. And I should tell you that everything that he did this way. We also tried it with the 75th percent, with the 0.75 and the 0.8. It always comes out the same. And then he did a very simple manipulation. He just took these trials and he nudged them over. So instead of being oriented on the 0 time point when the taste was delivered, they're now oriented on the time point that that state became maximal. And so same data set, the average movement of these firing rates was about 150 milliseconds, although some of them had to move, some neurons had to move, some trials had to move a lot. But now when we do the exact same analysis, we've got this time point centered in the analysis, the time point at which the state comes on. And when you do that, suddenly your transition to palatability is super fast. And so this transition happens this fast at the 0 time point. And we should tell you that there's a lot of stuff that goes in here. And in my old talk, I spent a good deal of time pulling apart this result. I'm not going to do that now because I have a lot of new data that I want to get you to. And so I'm going to ask you to, for now, take on faith that we've done a shit ton of analysis on this. And it's not just that that transition is fast. That transition is effectively instantaneous. We cannot figure out a way to make it faster. In fact, we did dummy data with simulated firing that was made from our original data. And the only difference is we actually made sure that the transitions were instantaneous. We got exactly the same curve because you do get some smearing from the way you do analysis. As far as we can tell, palatability comes on in a moment. The great thing about that for us is that now we have a single trial measure. We can use HMM on single trials, and we can tell you when that transition occurs on single trials, which means we can get to the question that was asked, which is how does that relate to individual behaviors? And here's how it relates. So we basically did, I'm going to reduce this part of the data to the single most important plot, which is a scatter plot on which one axis is the latency to gaping. And on the other axis is the time of that transition to the palatability related state. And,
One trial.
yes. So I'm happy to talk about this at length. We, at this point, are doing this with about 75 neurons per ensemble. We've checked. Yep, but we've tested this. We can do this with 6 neurons. We can do the same thing with 6 neurons. And I'm happy to debate about this. When you hear about how important it is, we got to get more neurons and more neurons and more neurons. This activity, this dynamic that I'm talking to you about is not sparse. This is what the brain is doing, we would argue. You don't need a bunch of neurons to see it. We're recording from a bunch of neurons now because everyone's really impressed. But you can see exactly this pattern with 6 neurons, and it doesn't change when you have 75. And so what is that? So here we have the diagonal line in the middle of this plot, which means that anything that falls on this line, the behavior occurs at exactly the time of neural transition. And I should mention that we're using EMG, electromyography, to report the behavior time, because with a freely moving rat, it's hard to actually tell when the gape starts. And so any dots that occur above the diagonal line means the neural transition happened first. Any dots that happen below the line means the behavior happened first. And here's a full data set. And so what you can see is that with the exception of a couple of stray trials, which we probably just got something wrong in our analysis, the vast majority of them show that first of all, there is a huge variability in the behavioral latency and a huge variability of the time of neural transition, that the two are linked, and that in fact, reliably, the neural transition happens a couple hundred milliseconds before the animal gapes. And I can show you, I took out of the talk, again, for brevity's sake, just individual trial data. It happens again and again, no matter how early that transition is. A few hundred milliseconds later than that, the animal starts gaping, which means that with these data and this analysis, we can not only tell you what behavior the animal is going to do, we can tell you when the animal is going to do it. Okay, but that's all phenomenology. And so we move from there on to perturbation studies. Being psychologists, we started with a behavioral perturbation study in which my postdoc Jen Lee manipulated the behavior by cueing. And what she did was very simple. She cut down the data set she used, so that she was just using sucrose and quinine and just strong versions and dilute versions. Really, none of that matters. All you need to know is that the only thing that caused gaping is a very strong quinine. And so what she did is every time before she delivered strong quinine, she played a tone. And so across the session, the rats learned when strong quinine was coming, and that caused them to change their behavior. And so from the first half to the second half of the session, the latency to gaping reduced by about 150 milliseconds. And just so you know, when she didn't use the cue, there was no change from the first half to the second half of the session. And then she said, quite simply, okay, what happens to the neural activity? What happens to the timing of this transition? And it was almost magical. Basically, across from the first to the second half of the session, the latency to that transition also moved by 150 milliseconds. And so when you perturb the behavior itself, the neural activity is perturbed with it. But of course, what everyone also wants to know is what happens if you perturb the neural activity. And so my graduate student, Narendra Mukherjee, decided to look at this, and he looked at it with optogenetics. And so this is just a bit of brain tissue. This is showing where the injection of the virus with ARCH-T, which is an optical silencer, and GFP, which is of course fluorescence, were put into gustatory cortex. And then he let the animal recover, stuck in a fiber optics. And rather than doing the standard thing, since we seem to be constitutionally incapable of doing, decided to do it a little differently. And so he delivered 4 different trial types. In one of them, there was no laser at all. In another one, the laser was on for half a second at the beginning of the trial. And then there was a section of some trials in which the laser was turned on for half a second in the middle of the trial, and some trials in which the laser was turned on for half a second late in the trial. This, by the way, is a good way to avoid needing an empty virus control, because we're just comparing different kinds of laser delivery and not laser to no laser. And so there's a lot of really cool data in this study, but I really just want to focus in on this middle one, because the range of transition times and gaping times, if you recall, is somewhere around there. That's the huge variability in when the animal gapes. And so looking at Narendra's data, with the time of the GC, just looking at the time of GC perturbation being from 0.7 to 1.2, again, half a second. This is a really short perturbation. And in his data set, the average time of gaping was about 900 milliseconds. But there was, of course, a big variability in that, which left us with what we thought was a problem and turned out to be a wonderful thing, which is that sometimes on some trials, remember, all of these trials, as far as Narendra and I were concerned, were the same. We turned on the laser we gave the animal quinine at 0, turned on the laser at 700 milliseconds, turned off the laser at 1200 milliseconds for every one of these trials I'm going to talk about. But on some of the trials, the neural transition that I've been talking about hadn't happened yet at that time point, because there's this big variability. And we were doing recording, and so Narendra could identify after the fact the trials in which the animal had not made that transition, which GC was not yet doing palatability-related firing. And on those trials, gaping was massively delayed by over half a second. So basically, the animal, for all intents and purposes, wasn't able to gape until we turned the laser off. And then there were a few trials in which we missed it, in which the animal had already made the GC had already made the neural transition into palatability coding. And it turns out that on those trials, our laser had 0 effect. This looks like the laser had a negative effect, but that's just because we're only looking at the most early trials here. Basically, this had no effect, and we did our best to analyze this as carefully as we could. If the laser turned on 25 milliseconds after that transition, we had no effect. And so what that means, first of all, and this is sort of an aside, but I'd like to sort of bring it up. What happens when you inhibit GC, you might ask? The answer to that question is, well, it depends. And so, in fact, we might write, and in fact, we did write in an opinion piece first authored by my postdoc, Jean-Yu Lin, that the idea that you figure out what neurons do by knocking them out is simplistic and wrong. That what neurons do is determined by exactly when you ask them, and not just when you ask them, but what they've been doing just before. But more for the purposes of this talk, another point that carries us to the next level is that gaping did eventually happen. We were kind of hoping that by doing this, we basically just blow the animal's trials to smithereens, and he wouldn't gape. She wouldn't gape when we turned the laser on. That did not happen. We delayed it massively, but once we turned the laser off, the animal gaped, which means, well, it could mean a lot of things, but it probably means that something else is involved. Knocking out GC wasn't enough because there's a system that's actually moving you toward gaping, and once you let GC go, that system is still able to communicate with it, and the fact of the rest of the system drives the behavior home, which suggests that GC is maybe the output station, and that this signal, that is this neural transition, is the signal to the rest, to that, okay, we've reached the decision to gape, but that is not making that decision alone. Yes? So the stimulus is still in the mouth, although the animal is engaged in massive removal of it. And it's also salivating. And so we find that while you can still detect that the animal has not gotten back to ground zero, certainly you're not getting much in the way of behavior. And neural activity is receding down to nothing by about a second and a half. But so if GC isn't doing this alone, then what's going on? And so we focused on the basolateral amygdala, although at this point we've got about 4 different projects going on in the lab having to do with 4 different other brain areas. But why do we focus first on the basolateral amygdala? Well, for starters, it's reciprocally connected to GC. In addition, basolateral amygdala neurons are responsive to tastes, and not just responsive to taste, they're responsive to tastes on the same clock as GC. When you look at BLA neurons, of which this is one, and so this is again, the formatting is a little different because this is the work of my postdoc Abu Mahmoud recently. You can see, but we also did it, we did this way back in 2009 as well. You can see that there is an early epoch, which in this case of this neuron, there's no firing. And then you get a middle epoch, and then you get a late epoch, and you can see the responses change going into each of those epochs. And remember that in GC, that middle epoch is the identity epoch, and that late epoch is the palatability epoch. It turns out it's a little different in BLA. It's the same clock, but in the amygdala, the ability to tell what the taste is comes on with the identity epoch, but then goes off again. So basically, it's only briefly taste responsive. Furthermore, that brief taste responsivity is intrinsically palatability related. So when you do the palatability correlation, you see that this middle epoch in basolateral amygdala neurons is already giving us palatability information, is already giving us palatability related firing. I have to be careful with that. My closest collaborator is a mathematician, and he always tells me, you know, information is one thing, relatedness is another. And so what you've got here is you've got palatability related firing right off the bat, right in that middle epoch. And so in addition, finally, and I don't have time to go into all of the work that's been done by a lot of my colleagues, the basolateral amygdala is known to be involved in driving emotion-laden decisions and learning. And so the decision that you hate what's in your mouth is intrinsically an emotional behavior, and the amygdala has been shown to be involved in those. And so that's why we decided to look at BLA. And so my postdoc, Zhengyu Lin, decided to ask, to the degree to which BLA is important for the dynamics we see and what we see in GC. And so what he did was he infected BLA with ARCH-T. And so the BLA neurons all got that, produced that channel, and so did their axons. And so then what Zhengyu did was he put his optotrodes, so electrodes plus fiber optics, just into GC. And the idea was that with that optical stimulation, he could perturb activity not in BLA by itself and not in GC, which is not infected, but within the axons that are projecting to GC. I'll admit, didn't think this was going to work. We've done a lot of controls, can tell you that not only does it work, I'm going to show you the impact of it, but we've also done the controls to show that it's not actually messing very much with activity back in BLA, which we were worried about. But the important thing I'm going to show you is that when you do that, you mess with palatability in GC. About 50% of the neurons in GC that had palatability related firing lose it when you knock out that input pathway. And you know, and I'll come back to it in a second, that some neurons that didn't have it develop it. We're going to come back to that in just a second. Point out that this, one of the reasons we're pleased about this is that it actually replicates earlier work done with actual inactivation of the amygdala itself. And so that these data look remarkably like that. But we weren't super impressed, because again, it's not just that there's some palatability information that remains in GC. There are some GC neurons that when you block BLA input, get palatability related information. And so we don't really have a good answer yet for exactly where that information is coming from and why it's there. But I can tell you that it made us say, okay, what really is BLA doing then if it's not just blocking palatability from getting into cortex? And the answer is actually a little more interesting and has to do with the dynamics. When Zhengyu in his own data set looked at the onset of palatability response in relation to that transition, he saw what we've seen before, which is a very sharp rise. So again, as you go into that palatability related ensemble state, you get palatability information. And then he's asked what happens when the laser's on, and the answer is that. And so in fact, in looking at the entire data set, the slope of that rise of palatability related information is cut more than in half by the loss of BLA. And so BLA, yeah, it's involved in having palatability related information in cortex, but what it's more robustly involved in is making those dynamics happen. So without the BLA, you don't have those sharp dynamics which we've related to behavior. And you might ask, so what happens to the prediction of behavior? It goes to hell. Now, does it go to hell because the behavior goes to hell, or because our ability to use HMM goes to hell? We're not entirely sure yet. But my other postdoc, one of my other postdocs, Abu, decided, well, if that's the case, then it becomes reasonable to ask, if you need BLA to make these dynamics happen, and we already know that BLA works on the same clock, is it even reasonable to be talking about BLA and GC separately? Are they really part of the same unit? And so all he did was he put electrode bundles down into both structures simultaneously, and then he just did another recording session. And so what we did then is we looked at HMM solutions to the firing in both regions. And what we saw is that in general, we saw that when the GC neurons transitioned into the late state, so did the BLA neurons. And when on a different trial, that transition happened earlier. It happened earlier for both. When it happened later for both. And so we went and did the full analysis on the entire data set, thinking that transition 1 is going from the detection to the identity phase in GC. Transition 2 is going from identity to palatability. Transition 3, which we generally don't even do much with yet, is coming out of the palatability related state. And what he found is that there is a synchronization, a correlation between when this happens, but only for the transitions in and out of palatability. And so what he found is that GC and BLA are in fact coupling for purposes of doing that palatability coding. And so this is a paper that just got published just this past year in work that he's still doing now. He's gone beyond this because, of course, this correlation doesn't necessarily mean exactly at the same time. It could mean one leading the other. And in fact, when he does an analysis of exactly the relationship between transitions in the two, and in a plot like this, any example that's out here means the BLA transitions first. Any on this side means the GC transitions first. And for that first transition, which remember is distinctly uncoupled, BLA comes first. And so what we got at the beginning of this response is GC and BLA are doing their own thing. They're both getting taste-related information. It's getting to BLA first. But then look at the next two transitions. Somehow in between transition 1 and transition 2, the two not only become coupled in that there's correlation, they become coupled in that they're synchronous. In that by the time you get out to here, now BLA and GC have effectively become a single processing unit. They are now working together, and that they click into alignment. And mind you, I'm almost done now, and this is work. All the stuff that I'm showing you now is also unpublished, and so I'm happy to talk about it. Be lying if I said I understood it all, but that's where we are now, with one exception. So from this, Abu said, okay, what I think is going on, Don, is that early in this response, before they synchronize, remember that you've got palatability already happening in amygdala. I think that the amygdala is pushing the system into a coupled mode. And that what's going on is that information from the amygdala is what's being used to drive the system into a particular coherent global state. And so he went ahead and did some analysis using Granger causality, which for those of you who don't know is, sort of a rough and ready first pass at an analysis that says not just is there coherence between these two regions, but is there directional coherence? Is it fair to say that one region is influencing the other? And what he found is when he looked at the BLA to GC connection with the influence going from strong to weak, what he found was that the only notable coupling that happened after the taste hits the tongue is in a very low frequency range. And in fact, it happens exactly when we're going between transition 1 and transition 2. That during the period when BLA is coding in a palatability related fashion, before GC is, BLA information flow is going from the BLA to GC. In the meantime, by the way, this is not something that is reciprocal. In fact, GC, the influence of GC on BLA is typically is quite a bit stronger, and it is fairly stable. If anything, it looks like there might be a little bit of a decrease during that period, and then a reinvigoration, although again, these are very preliminary data, so obviously we have a lot of analysis to do before we can convince ourselves. But it seems like there may be, in fact, a situation where it seems like the BLA to GC flow is driving coupling, at which point the system emits a signal, and the GC then communicates out and says, okay, it's time to gape. And that's all I've got for you on data. And again, happy to talk about this, particularly because we haven't finished it yet. Yes.
I think this preliminary analysis has ruled out a third area that projects to both.
It clearly has not. It by the very nature has not. We are now doing experiments in which we knock out other regions at different time points, and that will give the proof. We think it's fairly safe to say that this is probably not from a common source, but we don't know for sure. But so my obligatory summary slide to tell you what I've told you, now that I've told you, GC taste responses reflect taste processing across successive epochs, which means those of you who have been wowed by simple stories of just some neurons, ooh, sugar neuron, some neuron, ooh, bitter neuron, it's just wrong. And in fact, ensemble analysis of single trials reveals that the onset of palatability coding is a sudden ensemble transition, and that this transition in turn drives behavior, probably by modulating a brainstem CPG. Another set of experiments that we're doing are looking into that right now. And I would like to do a shout out in particular. There's a lot of research from colleagues that I haven't really been able to unpack for you, but I would like to point out that one of my mentors is Eve Martyr, and she has spent her illustrious career, which has most recently resulted in her getting a big medal from the president, looking at a central pattern generator in the lobster and crab. And most of the work that she's done on that has been on the fact that there is a whole bunch of top-down modulation, which helps to determine exactly what that CPG does. We are actually thinking of the forebrain in those terms, that what it's there for, and we'd like to help Eve rescue modulation from the second-class status to which it has been resigned, that the forebrain is essentially modulating this brainstem CPG in a way that allows it to the forebrain to say, okay, you could go into gaping rhythm now. And finally, the mechanism of construction of this transition involves coupling between at least the GC and BLA into a functional unit, where it basically where it transitions synchronously in and out of the important state. And so that's where we've gotten into it now. And so I send you greetings from the Behavior, Learning, and Electrophysiology of Chemosensation Lab at Brandeis University. This is, I think, a complete list of my people. I'm not entirely sure, but every single person who is underlined here contributed a first author paper to the talk I just gave you. I should also give a shout out to my most frequent of many collaborators. Brandeis is a very incestuous place. We're all working in one other lab, but my most frequent collaborator is Paul Miller. Paul is a recovering physicist and a brilliant modeler, and I can barely add. And so he is the person who makes sure that all of our computational work actually can be trusted. I would also like to thank, of course, funding from the NIH, the NSF, and the Schwartz Foundation for Computational Neuroscience. Thank you for listening. I'm happy to take any questions.
Oh, okay. And there's good time here for questions and discussions.
Hi. Thank you so much.
So this, your focus on whether it's sudden or gradual, this just reminds me of the whole my child. Yeah. So I want to know, there, of course, they want to say whether it's an accumulation of everything. So what does it mean in this case?
Right. That's a great question. The paper, Sadaka et al. 16, fairly simple paper. Excuse me?
Do you repeat that question?
Oh, I'm sorry. The question was that it was noticed that when we talk about sudden transitions, we are sort of Mike Shadlin is suddenly in the room, who talks about the neural basis of decision making in primates, and has talked for years about a biased random walk and evidence accumulation, which necessarily leads to a ramping of activity. And he's talked about that at length. And so the 2016 paper that I was referring to at that point took a year and a half extra to get published than it was supposed to. And I should mention, as I've mentioned before, I don't send papers to Neuron because I got no interest in a 2-year turnaround. That's one of the reasons. Usually my papers, between my initial submission and the publication of that paper, is 6 to 8 months. And this took a long time. And the reason was because we ran afoul of the Shadlin people. And so in that paper, stuff that I'm not talking to you about to you this time, is we went to the wall and beyond to show that you cannot explain this with accumulation of information, that this is an intrinsically attractor network model that explains this. We also have Paul and I wrote a 50-page paper, computational paper. If you're having trouble sleeping, I'll send you a copy, in which we show that the Shadlin model doesn't always give you the best result, and in fact, the kind of model that an attractor model that we use that's noise driven can be more optimal. But I will say that when I gave this talk at Columbia, Mike and I spent about an hour and a half in our half-hour meeting talking about different behaviors that might call for different types of decisions. And I'm pretty comfortable with that answer, that in fact, the kind of task that Shadlin does is a task that benefits from accumulation of information, not so much this one. Having said that, it's worth noting that I also gave this talk way before we got it published at a computational meeting where Jonathan Pillow was hanging out. And Pillow went back to his collaborator, Huck, who used to work for Shadlin, and the two of them took apart Shadlin's own data, and they concluded that you don't see actually a random walk there either. And so we think that at least in our system, it's an entirely different network.
Same with amygdala, I missed that part of your talk.
Yes. And in fact, we can only say that recently because here is a place where more neurons mattered. Amygdala is a harder hit for those of you who are forced to try to do it. We've only recently been able to get enough neurons to say that yes, the same is true there. Yes.
You talked a little bit about how many neurons do you need to make this estimate, and of course it depends on how many neurons are transitioning together.
Right.
And I think you just were saying that in the amygdala, it's not the same scale, so you need more neurons. Is that right?
So in the amygdala, the problem was that we were generally getting fewer neurons always from amygdala than cortex, and so it was only with I'm a technophobe personally, and so it was only with my graduate students forcing me into the modern age that we got to a point where we were actually getting a substantial number of neurons simultaneously from the amygdala. It still takes more because one of the things that matters is firing rates, and so the amygdala responses amygdala neurons tend to be quieter than cortical neurons, at least the BLA neurons in the awake animal, and so the firing rates that we were working with, the firing rate transitions were smaller transitions. And so it took more neurons there for that reason as well. Yes.
Neurons that suddenly turn on, and also the neurons that suddenly turn off.
Mhm.
Are those the excitatory neurons?
So it's a mix. We actually, so with our technique, we cannot definitively show what neuron types we're recording from. We can only do putative, and we could go in and infect interneurons and do that. And to be honest with you, we just haven't felt it was urgent enough to do that. But we have done putative pyramidal cells and interneurons, which you can pretty well identify on the basis of basal firing rates and the width of spikes. And what we find is that we do have neurons in which the transition is down and neurons in which the transition is up, and that does not obey neuron identification at all. That we get both interneurons going up and pyramidal cells going up.
Yeah, the reason I was asking is it's kind of hard to get neurons to all of a sudden suddenly transition from silence to firing a lot.
wondering if it's something local where there's like burst of excitation or release of inhibition, or if it's something external.
Yeah, so don't know. The honest truth is just simply that we haven't been doing experiments that are designed to get at that level of analysis. I can tell you that we see them a lot, and that these sudden transitions into high firing rates are the most common thing we see. And wouldn't surprise me at all if it's happening because of the removal of inhibitory neurons. Don't know. Have to do a kind of experiment that we generally don't do in our lab to really know the answer. Yes.
Range of causality preliminary data, slow, what you call slower oscillations, those to me looked kind of theta-ish.
Ooh.
Is theta something that's playing a role here? I mean, obviously it's playing a role in olfaction and the sampling of smells. I don't know about.
Yep. And so there's a lot of rhythms going on up there. It's a little tough to pull them apart because some of them are driven by behavior. But we definitely have seen high theta in our activity, and haven't and you know, these data just came in, and so haven't really taken that apart at all. But certainly it looks like what we're seeing from BLA to GC is either high theta or low alpha. Yes.
Hey, I was interested in the variability in the time, like onset of the palatability phase, and also the data you were showing about the animal to animal variability, and whether or not they even gave or not. And I was curious whether or not there's anything in the early part, you know, the earlier kind of two phases that may be predictive of either the onset of timing of that palatability phase or the likelihood that they gave it all.
We have not found anything. So the question is, can we tell in a particular trial at the beginning whether it's going to be an early transition to palatability or late transition to palatability? We've been trying everything we can think of, and we can't find anything yet. Now, I'm sure it's there. Of course, actually, it doesn't necessarily have to be there. One of the things that can drive a system like this is noise. And in fact, I will say that that's one of the things we like about our system better, about this network's better than an evidence accumulation network, is because those networks, basically any noise is bad, and these kind of networks actually thrive on a certain level of noise. But we've been looking like crazy for that stuff, and we haven't found it yet. Having said that, I don't want to say it's not there. I think that we haven't had enough creativity and insight yet. I got a brand new postdoc whose entire job is to search for that. And so check with us in about a year. Yes.
So I had a question about the sensory neurons themselves, and if you record their dynamics, whether you get a similar kind of bistable state switching. So if you taste something, I can imagine that there's a commitment, like in the tongue itself, right? To taste more, taste less.
It's a great question. And so I don't even ever present the taste circuit anymore. But the primary gustatory cortex is actually several synapses north of the tongue. You go from the tongue to a medullary nucleus, to a pontine nucleus, to the thalamus, to the cortex. And so we haven't gone back all the way to the sensory neurons. We have, however, gone back to the very second relay, which is the pontine relay. And I have a postdoc who nearly destroyed herself getting good neurons down there in the awake animal. And what she found was almost exactly what we see in cortex. Now, we need to do a lot of work on that. And it was just, she basically just came back this close to quitting science. And so we haven't been back. But what I can tell you for sure is that processing is at least engaged in that is at least as early as the pontine second relay. Yes.
You said you think imaging is too slow for these kinds of, to get the kind of data you need. Would you include like newer voltage indicators in that camp?
No.
Getting like up to kilohertz sampling?
The question is, in my callous dismissal of imaging, am I ignoring the new stuff? And the answer is, I pretty much am. it's important to note at this point that a couple of my best friends are imagers, and in particular, I'm very close with an olfactory imager named Matt Wakowiak at the University of Utah, who's amongst other things a brilliant technician, and he is one of the people working on making the newest generation of voltage indicators work. And no joke, if it gets to the point where the details have been ironed out, again, I'm a technophobe, I don't want to develop anything. I want to use things to answer questions. As soon as they've got that to a point where I can see this time of activity in the firing, I'm going to drop electrodes like a hot potato and go to imaging then. Mostly I'm talking about calcium imaging, and I've had friends who are doing calcium imaging in cortex, and it's pretty clear that they can get a shadow stuff, but the signal between the signal noise ratio being relatively low and the slowness of the calcium changes themselves, they can't really get single trial resolution yet.
So we still officially have 1 minute, so I'd like to ask you, this might be a pretty open-ended question. The behaviors you're describing are based on the gaping mostly in this talk. I'm just curious, if the animals were, this may not be as naturalistic as that, but if the animals were trained to discriminate between very subtly different whatever you imagine, could you imagine forcing decision-making in this time frame to really get at the question of like when the information is available?
And so that's a great question, and we've devoted a couple of studies to that I haven't talked about. In one of them, we did almost exactly what you described. We did. There's one of the tried and true tasks of the behavioral neuroscience is the 2 alternative force choice task. And so we did a 4 alternative force choice tasks so that we could get rid of palatability as a signal. And so a palatable and unpalatable tastant, meaning respond over here to this lever over here, a palatable and a different palatable and unpalatable tastant, meaning respond over here. And what we saw is we basically pushed the decision variable way up early into the identity epoch. And at the same time, we've also done conditioned taste aversion, in which we basically swap a taste from being palatable to being unpalatable in a single session. And with that, we see nothing in that middle epoch, only changes in that late epoch. And so we can do it. And probably we should be doing a lot more of that. But that's all we got so far.
All right. Well, let's thank Don for a great talk.
Thanks, everyone.
The sandwiches were stealthily brought in. Please partake.